No, class.
Yes, ma'am. Yes, ma'am.
Okay, so then what happens is that once the cells are buried, embedded in these agarose plugs, then what you do is all the operations of cell lysis, all the operations of isolation of DNA, they are performed while the cells are embedded in the plug itself. The cells will not come out, so you do not require a pipette. So what you do is that all the steps, for example, agarose plugs are prepared. You take out these agarose plugs because they are now solid. They would be like this gel, right? So they are now solid. You can take this plug out, right? And then what you do is you take a glass tray and add lysis solution, right? So lysis solution, for example, for gram negative bacteria and for gram positive bacteria, what is the lysis solution class? I think students have left already. The number is less and those who are there are also gone, but they are there.
Okay, sir.
For gram
Okay.
negative, that is SDS detergent. We add
Yes.
detergents.
Right, right. So you're going to add these lysis solution, for example, for gram negative TriCl, EDTA, and then you can add SDS as well. And for gram positive, you're going to incubate it with enzyme and then add TriCl, EDTA as well. So what happens is that when we put this solution in glass tray and add agarose plugs, so this lysis solution will penetrate inside the agarose plugs, and therefore the lysis will happen while the cells are embedded in the plug itself, right? So after that, all the steps, so for example, when you want to treat it with proteinase K or RNase A, all these are performed while they are there in the plug itself, right, class? And once you have performed this process, then you do washing so that all the cellular debris, so you do not treat with phenol and chloroform because phenol and chloroform will actually affect the mechanical stability of agarose. So you can perform lysis while the cells are at agarose. You can treat it with proteinase K, you can treat it with RNase A, and then what will happen is you will incubate it with TrisCl again, do a couple of washings just to remove the cellular debris, and then these agarose plugs, class, listen to me carefully, these agarose plugs are now placed carefully with the help of a forceps into the wells that are created, right? So for example, the wells that are created here, right? So instead of using a pipette tip and adding the DNA mixed with bromophenol blue, what you will put is agarose plug. What you can do is, if you want, you can incubate in the glass tray, you can add bromophenol blue and then incubate the agarose plug with bromophenol blue, and then put the agarose plug on these wells, inside these wells. So that means the width of the well should match with the width of the agarose plug, so that the agarose plug just fits inside the well. Okay, so this way what you are doing is you are not using any pipette, number one, you are not using any centrifuge to concentrate the cells. Whatever cells are there, you are using as it is. But then just to increase the number of cells, you allow them to grow overnight and then you separate them. Now I'll show you how the separation is achieved. So this is the paper which talks about pulse field gel electrophoresis. This is available. If you type in Google, you will get this paper. Estimation of chromosome number and size by pulse field gel electrophoresis in Candida species. This is how this pulse field gel electrophoresis
Ma'am,
differs. Yes.
I was asking like how do we know like if we don't count it, then how can we know like how many numbers, chromosomes numbers?
Yes. So that I'll show you in a second. Right.
Okay.
So look at this figure. So here what you are seeing on the left is pulse field gel electrophoresis, and what you are seeing on right is conventional electrophoresis. So in pulse field gel electrophoresis, you see that electrodes are present at three places. So they are present in diagonal direction, in this direction, and in vertical direction. Whereas in conventional electrophoresis, electrodes are present only and only in vertical direction, right? Now, the term that is used is called pulse field gel electrophoresis. So the reason it is called as pulse field gel electrophoresis, gel because you are using like the agarose gel, the same agarose gel that we discussed, but we are using pulse field. So pulse field means you are giving a pulse in one direction and then switching it off. Then you give a pulse in, let's say, vertical direction, switch it off. Then you give a pulse in again a diagonal direction, right? So what happens is that when you do these pulses, so the distance traveled becomes different. It's like displacement. So you have in this direction, then in this direction, and then again in diagonal direction, and then again straight, and this cycle can be repeated. So as a result of this, the distance traveled by the molecule increases actually, and this results in better separation. So now to answer the question which Ratnesh you were asking, like how can we determine the number? So the number is determined by two ways. Number one, if you have a cell which contains two differently sized chromosomes, you can actually run it, and then you will see that even though the differences are in megabases, for example, Vibrio cholerae has 3 MB chromosome and E. coli is 4.6 MB chromosome, you can separate that. If you have a situation where E. coli is 4.6 MB and let's say another strain, Burkholderia, is like 4 MB, that means a difference of only 0.6 MB. Still, you can separate. So that means if the sizes are different class, we can separate by pulse field gel electrophoresis. Is it clear, Ratnesh? Is it fine?
Yes, ma'am, it is clear.
Okay. So then, and the idea of PFG is clear to you?
Yes, ma'am.
Yes, ma'am. Yes, ma'am.
Okay, so maybe now I'll show you the video for this one. So PFGE is not there in our department, not there in UG lab as well. So you will not get a chance to see this, but then so this is from. Can you see class or no? Can you hear the voice sound?
Yes, ma'am.
So could you hear the sound also?
Oh, not right now, but I think we can.
Okay. 1 second.
Pulse field gel electrophoresis,
Can you
or
hear
PFGE,
students?
is
Yes,
the
ma'am.
Yes, ma'am.
separation of very large DNA molecules. Under standard electrophoresis conditions, an electric field is applied in one direction on the agarose gel, and DNA molecules larger than 40 kilobases essentially move together through the gel, independent of their size. In contrast, PFGE applies an electric field that periodically changes direction. By introducing an alternating voltage gradient on a PFGE system, very large DNA fragments can be separated by their reorientation and movement at different speeds through the gel. Larger pieces of DNA are slower to align their charge to the change in direction, while smaller pieces are quicker to align, resulting in greater separation. let's take a closer look at the equipment and conditions that will lead to successful PFGE. Here is the electrophoresis chamber, which contains the running buffer. The agarose gel should be well anchored in the middle of the chamber so that it doesn't float around. There are multiple pairs of electrodes around the gel, set in a hexagonal grid. This hexagonal shape of electrodes allows for the directional switch of voltage. The voltage is periodically switched among 3 directions, one that runs through the central axis of the gel, and 2 that run at 60-degree angles on either side. The PFGE technique takes longer than standard gel electrophoresis due to the size of the DNA fragments and because the fragments don't move in a straight line. To prevent the agarose gel from overheating due to the extended period under the electrical field, which could result in a loss of resolution, a cooling module adjusts the temperature of the buffer. A typical PFGE program is run at 14 or 15 degrees Celsius. It is important that no air is left in the tubing to and from the cooling module. On the control module, the migration protocol can be entered and monitored. This includes the initial and final switch times, the duration of the migration, the angle of electrophoresis, and the voltage. The larger the DNA fragments, the longer the pulse times should be, and the longer the total migration time. The switch time is the length of time that the electrical field is pulsed in a single direction. For example, a 10-second switch time means that the electrical field will be pulsed in one direction for 10 seconds and then switch to another directions for 10 seconds. Samples with a wide range of DNA fragment sizes, such as NEB's PFG markers, can be resolved by ramping up a short switch time to a longer one over the course of the run. Most PFGE protocols are optimized for 6 volts per centimeter gel runs and optimized for a 120-degree angle. With any PFGE system, depending on the migration protocol, you can separate DNA fragments or molecules from 10 kilobases to 10 megabases. Here are separation examples of the lambda monocut mix from 1.5 to 48.5 kilobases, and yeast chromosomes from 225 to 1900 kilobases.
Followed class?
Yes, ma'am.
Yes, ma'am.
Okay. So class, can you see the slides?
Yes, ma'am.
Okay, so this is the link I have pasted over here. So now for separation of chromosomes, we are going to use pulse field gel electrophoresis. For small plasmids and for smaller DNA fragments, you are going to use the conventional horizontal agarose gel electrophoresis. But then what about oligonucleotides? So we discussed in the class that for oligonucleotides, you require polyacrylamide gel electrophoresis. So that means the pores in polyacrylamide gels are smaller or larger than agarose. So have you studied polyacrylamide gel electrophoresis class?
No, ma'am.
No, ma'am.
Okay. So all of these was never taught in biochemistry or SPL? No, never.
No, ma'am, I don't
Ma'am, we
think
did a
so.
bit in biochem.
Yeah, ma'am, we did just gel electrophoresis, not specific types of it.
Okay, but then you forgot everything. Is that true?
No, I mean, we haven't done specific types of gel electrophoresis. We have done just gel electrophoresis in
But I
general.
thought it's important to
No,
know
ma'am, I guess we did a bit of SDS page, like,
for proteins, right?
yeah.
Okay, so then that part we'll cover. So polyacrylamide gels are actually made up of monomer, which is acrylamide, and then we also use a cross linker, which is bisacrylamide. Okay. Acrylamide is nothing but 2 acrylamide molecules, right? 2 acrylamide molecules which are linked by a methylene group is called bisacrylamide. So acrylamide and bisacrylamide are used to make polyacrylamide gel. But then the monomer and cross-linker, when they form this together, when they form polyacrylamide, this formation is not like a simple reaction. This is actually a free radical assisted. Okay. So when we say free radical assisted polymerization, there are two chemicals. One is called as a catalyst, right? And the other one is called as an initiator. So in this case, class, the catalyst that is used is called TEMID, and the initiator that is used is called ammonium per. So what happens is that this initiator, so the name initiator is given because it is the initiator which helps in the initiation of the reaction. So in this case, ammonium persulfate is the initiator, the catalyst is TEMID, and then the monomer is acrylamide, the cross-linker is bisacrylamide. All these components have to be there together. And then this TrisCl can have varied pH. So the pH of the TrisCl varies depending upon the requirement, which I will be telling you depending upon the gel types that we use. Okay, so now I'll show you how exactly the gel looks like. I think I have it here, right? Yes, so this says that the polymerization is initiated in the presence of BIS is the cross-linker. This is the full form of TEMID, N-N-N-N-N tetramethylene diamine. I think I had, yes, this is the figure. So here, this is how the polymer is formed. So you have acrylamide, you have bisacrylamide, and I told you that two molecules of acrylamide connected by a methylene group is bisacrylamide. In the presence of persulfate ion, you will have the polymerization of the gel. Now acrylamide students, acrylamide is neurotoxic. So when you are actually weighing the acrylamide, even a fine dust can be dangerous. So therefore you need to wear mask and do these measurements very carefully. Usually we do not give this to UG students. Usually PhD students prepare the solution. So once it is in solution, then it is fine. It is not that dangerous. But if you by mistake, if you inhale the powder or even dust, then it's very, very dangerous. So you need to be very, very careful. So this is how the free radical is generated with the help of TEMID. So TEMID catalyzes the decomposition of the persulfate ion to give a free radical, and then it's a chain of reaction which results in the polymerization of the gel. But again class, in polyacrylamide gels also, we can actually create gels with different percentages. So for example, gels can be created which are 6%. You can create gels which are 12%. You can create gels which are 10%, and you can go maximum up to 20%. Right? So again, 20% gels will have smaller or bigger pore size?
A smaller pore size.
Pore size. And 6% gels will have?
Bigger pore size.
Bigger pore size. So for example, if you have a sample which contains 100 base pair to 500 base pair, 100 base pair and maybe 500 base pair, and then for example dinucleotide, then hopefully the class can typically tell me like which percentages are we going to use. So usually
120 for the 100 base pair.
Right. So 6% gels are usually used for 100 to 500 base pairs. You can use 6% to 8% gels. But when we talk about using polyacrylamide gels class, we can run we can perform DNA sequencing reaction. So when I will be teaching you sequencing by Sanger's method, so nowadays fluorescence based methods are used. So I'll give you the details when we will discuss DNA sequencing, but the earlier methods involved use of polyacrylamide gels only. So when we perform Sanger sequencing reaction and when you use radioisotopes for identifying the nucleotides, then in this case the gel percentage that is used is 20% gel. So with 20% gel class, we can separate single nucleotide differences. So for example, if you have an oligo, let's say if it's a dinucleotide and the other one is trinucleotide and the third one is tetranucleotide, you can separate in a 20% gel.
Oh, but ma'am, how can we see like a dinucleotide that will be very, very smaller? How can we see that?
Right, so for observing that, what we do is that we use radioisotopes. So that is why I told you that when you are performing the sequencing reaction class, so the earlier methods when Sanger discovered the technique, he was using radioisotopes. But nowadays people use fluorescent tags, fluorescent dyes. So you observe these bands with the help of radioisotopes. We will be discussing this in more detail when I'll be teaching you DNA sequencing. But again, because of radioisotopes, you can actually capture the image on an X-ray film. So what happens is that you run the sequencing gel, which is like 20% polyacrylamide gel. You separate the DNA, I mean separate the oligonucleotides, and then you expose it to X-ray film. That means you place the X-ray film in dark and then you develop the X-ray film. And this developing process is very similar to the conventional X-rays that you get in hospital. Is it making sense or no?
Yes, ma'am. Yes.
But again, we are going to discuss this in a little more detail when I'll be teaching you sequencing. But just to give you an idea that if you are performing if you are interested in separation of single nucleotide differences, then you go for sequencing gels. But for bigger fragments like 50 base pair, 100 base pair, 500 base pair, you can use gel percentages from 6% to 12% to 15%, depending upon the requirement. These gels are performed in a vertical assembly and therefore this is called as vertical gel electrophoresis. So now the question is why we do not perform polyacrylamide gel electrophoresis on a horizontal assembly? Why at all we require a vertical assembly? Right? I mean, if the assembly is there for horizontal gel, why not prepare polyacrylamide gel on the same? So based on what I have explained to you just now, can you think of a reason?
No, ma'am. I don't think so. Like there will be gravity. Nothing else more than that. More like.
Nothing else, nothing other than that. Jaskin, Ayush, Jaskin Singh, are you there or no? Ayush, are you people there or left?
Mom, it moves down the pores, right? So like it doesn't use any electric field. So like based on the size, it will move down the pores, and like the smaller chains will move down even
Oh.
more than the
Also, the
larger.
pore size are small, so
What did you say, Ayush?
the pore size are small, so they will move.
Our sizes are small. No, I said something based upon what I explained to you just now. Dhwani, Ashi, Devpriya. No? Nobody wants to even attempt. Okay, so
This is a very wild guess. Could it be possible that maybe the gel, also the gradient of the gel sort of varies when we're at the lower end and the upper end?
No. So you people are thinking too complicated things. The reason is something very simple. So vertical gel assembly looks like something like this. Okay. So for example, you have two glass trays, right? So can you see this at the bottom right? You see the vertical gel apparatus with the red and the black knobs. Can you see?
Yes, ma'am.
Right, so these are two glass plates and then you pour the gel in between the two glass plates. So the reason we perform polyacrylamide gel electrophoresis almost always on a vertical gel assembly is because this polyacrylamide gel electrophoresis, as we discussed in the class, it is a free radical assisted polymerization. So what happens is that you need to protect it from light. So when you pour the gel on vertical gel assembly, so ultimately in order to polymerize it, you need to overlay it with isobutanol or something just to prevent any exposure of air. Okay, because this air can prevent the free radical assisted polymerization. Whereas when we run it on a horizontal gel, then the exposure is too much. So therefore the polymerization of the gel will be extremely difficult. Whereas in vertical gel assembly, if you just overlay it with a thin film, then polymerization is at least better. Is it making sense or no?
Yes, ma'am.
So therefore, when we talk about polyacrylamide gel electrophoresis, almost always we run it on a vertical gel assembly. And when we pour the slurry, you need to overlay it with isobutanol so as to facilitate the polymerization. So this is so much affected by free radical that even if your APS, so ammonium persulfate solution, needs to be freshly prepared, even if this APS solution is like a week old or a few days old, then usually you will find that the efficiency decreases. So it needs to be freshly prepared every time you take a powder and prepare it in a small Eppendorf tube and then use it afresh. Otherwise, you will see that the polymerization will be affected. So class, we are going to use this polyacrylamide gel for the separation of oligonucleotides. But then before I explain you, I would also like to tell you that this polymerization can also be assisted with the help of light. So instead of TEMID, you can use photopolymerization, and instead of ammonium persulfate, you can use riboflavin. So photodecomposition of riboflavin generates a free radical that initiates the polymerization. So now the question is, why at all, when you have APS, when you have TEMID, then why are we interested in photopolymerization? So photopolymerization is required under specialized conditions when you are separating these bands in a microfluidic device and you observe it under a microscope. So in this case, you do light assisted polymerization just to avoid the minimum exposure to dust, because see, when you want to observe the DNA under a fluorescence microscope, or right, so in this case, even if there is a little bit of dirt, that is going to fluoresce, and then the the whole purpose of observing it under a microfluidic device will be defeated. So what we do is that we do the polymerization while on a glass slide or on a microfluidic device, and then you allow it to go for photopolymerization in the presence of riboflavin. Okay, is it clear or no?
Yes, ma'am.
Okay, so then the question is, what happens when you have two fragments of the size 100 to 500 base pairs, and they are of same size. So tell me, how do we separate them? Or maybe I'll write 2 or more. So you can have a situation where you have 2 or more DNA fragments of same size and you want to separate them.
Ma'am, I think the size is same, but the composition will be different. So on basis of that, we can separate them using any technique.
Right, Deepak, very good. So size is same and you can separate it based upon the composition, and the composition is A, T, G and C. Correct. So what happens is that depending upon, so class, listen this part carefully. So depending upon the GC content, right? Depending upon C content, the DNA can denature differently.
Yes, ma'am.
Yes, so this feature is utilized. This feature is utilized for the separation of DNA fragments of same DGGE, which is also called as denaturing gradient gel electrophoresis, right? Denaturing gradient gel electrophoresis. So in this case, the gel that is used is actually polyacrylamide gel only, right? But what we do is that we use a denaturant. So the denaturant that is used is actually formamide, or you can also use urea. So urea or formamide are the denaturant, and then the word gradient is there. So what we do is that we prepare a gradient of denaturant. You are getting my point. So you prepare a vertical gradient. So let me use the term vertical. A vertical gradient of denaturant, which are formamide or urea. So you imagine a situation that you have a vertical gradient of these denaturants. So as a result of which, for example, you will have at the, let's say at the upper layer you have, let's say, the lowest concentration of the denaturant, and then you have a gradient, and at the bottom most you have the highest concentration of the denaturant, something like this. Is it making sense or no?
Yes, ma'am. Yes.
Right? So can I explain you this, or is it getting too much?
You can explain. We can.
Okay, just this part, and then maybe we'll discuss in details more. So this is the device which is called as DGGE, and this requires a gradient maker. So what you see on the top left, right? So this is a gradient maker. So here you have two different containers. One contains low concentration solution, one contains high concentration solution. They both are placed in a magnetic stirrer and connected with a tubing, right? This is the peristaltic pump, and this is the silicon tubing, and this is your vertical gel assembly. The glass plates are here, right? So what happens is that a gradient can be formed by mixing the two solutions, right? With the help of a peristaltic pump, you can control this mixing. So the low concentration would mix with high concentration, and ultimately you will pour it in this. Right? This is the real assembly, which looks like this, and ultimately you will create a gradient. The gradient is of the denaturant class. Don't forget, the gradient is of the denaturant. Okay, so the idea that is utilized in this technique is that when you separate two differently sized DNA, right? So depending upon their GC content, they will require different amount of denaturant for denaturation. Is it making sense or no?
Yes, ma'am. But
Yes,
how
ma'am.
can they separate like two DNA fragments different at different places? Like.
Right. So because you have a gradient, so for example, if you see in this figure, you have at the top low denaturing concentration, at the bottom high denaturing concentration, right? The bands are of equal size. When you load the DNA is negatively charged, it will move to anode, right? But depending upon the concentration of the denaturant, the mobility will vary. So, and this variation will actually depend upon the GC
Ma'am,
content.
ma'am, but mobility will not depend on like denaturants.
The
That will depend.
mobility will depend upon the GC content of the DNA, and the GC content of the DNA will actually be governed by the denaturant, because the moment, so for example, let's say you have a 500 base pair sample, 500 base pair DNA fragment, two DNA fragments of 500 base pair. One contains 60% GC and one contains 40% GC, let's say, right? So now please tell me that 40% GC will denature first or 60% GC will denature first. Then
40%.
40%.
very good. So 40% GC will denature first, and let's say it will require X concentration of formamide, right?
Yes,
Yes.
ma'am. Yeah.
Now 60% GC cannot denature at X concentration.
Yes, ma'am.
So then it will move further, and then when it will find the higher concentration of formamide, it will denature. That is appropriate for its GC content. Is it making sense?
Yes, ma'am. I understood. Like after denaturation, this DNA slows down, like
Yes,
goes
yes.
slowly.
Right, right. But then I'll be showing you a video and we'll be discussing this in a little more detail. What we do is that when we denature this, then if it's a double-stranded DNA, then actually the two fragments can completely fall apart. Right. So in order to protect that, what we do is we use what is called as GC clamp. So we are going to discuss this in the next class, what is GC clamp and how the separation is achieved. But I hope the basis is clear to everyone.
Yes, ma'am.
Anush,
I think.
Deepak, Jaskin.
Yes, ma'am.
Yes, ma'am.
Okay,
Yes,
very
ma'am, it is.
yeah. And if you have doubts, we can again discuss in the next class. Okay. So maybe I'll close the meeting.
Yes, ma'am.
Okay.
Thank you, ma'am.
Okay.
Thank you,
ma'am.
ma'am.
Thank you, ma'am.